Okay, we are going to do the first part of this chapter. Um, all of you going into nursing, medical, PA, are going to be taking a nutrition course. Of course, the dietetics people are going to take a ton of it. So we're not going to go into it as much detail as the book. All right, so a little bit of overview. These are reminders. You've had this in general biology. You had it in A&P1 a little bit. Chapter 3. Chapter 3 has a nice overview, simple overview of metabolism. Anabolic reactions. Building requires lots of energy input. Energy. Anytime I see energy, say energy, I'm talking ATP. Catabolic pathways and reactions. Energy producing, breakdown, degradation of molecules. You're going from large molecules to small molecules. Breaking of bonds releases energy. Anabolic reaction. You're taking small molecules, you're putting them into big molecules. Requires lots of input. Okay, you're looking at a brown cell down here at the bottom. This is a cell. And I wanted to point out that a cell is going to use the absorbable molecules that we ended up with, no pun intended, in the small intestine, went in blood. The amino acids, the lipids, the monosaccharides, by diffusion, went into the blood, carried by blood to the cells. Okay, it's at a cell. Now, the cells are going to then use monosaccharides and fatty acids, blah, 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 to build new things like cell membranes so that the cell can divide. Okay, so you go towards the anabolic side. But also the cell can use glucose and individual fatty acids to break down even further in aerobic respiration or anaerobic to produce energy for cell work, like making cell membrane, dividing, blah, blah, blah. All right, so those are catabolic reactions. We're going to be talking about aerobic respiration. We're going to be talking about lactic acid fermentation, which is the anaerobic route. The point here is the cell has to have energy from catabolic reactions to derive anabolic reactions. By the way, NRG means energy, ATP. When a cell breaks down a molecule in respiration, aerobic respiration, only about 40% of the energy in that molecule is turned into energy, ATP. The rest of it is heat. At the end of this semester, at the end of this chapter, we're going to be talking about heat control. Remember, that's where the heat's coming from. Okay, reminder about reactions. Electrons. Let me remind you of something here. A hydrogen atom is equal to 1 electron, negative charge, plus 1 hydrogen proton. I'm going to put in parenthesis P. That's a proton. Remember, hydrogen has an atomic number of 1. It has 1 proton in the nucleus. It has no neutrons. It has 1 electron in its orbit. That's it. So there's the sum total of hydrogen. So the electrons and hydrogens are moving together. They're kind of interchangeable. I mean, they're not exactly the same thing, but when I'm talking about passing off of electron, I'm talking about passing off of hydrogen, protons. Okay, so if a molecule picks up this electron or the hydrogen, it is said to be reduced. A molecule that gives it away is oxidized. Oxidation means to give away, donate. Electron, hydrogen ion, hydrogen proton. Reduction means to gain. The guy that gives it up is called a donor. The guy that picks it up is called a recipient. Now, there are some middlemen. They are the guys that pick it up pick up the hydrogens or the electrons from molecule 1 and give it to molecule 2. They are temporary electron carriers. You know one already. NAD is one. And there's a lot of enzymes involved in all of this. Each chemical reaction requires a different enzyme. Not going to get into the names of the enzymes. NAD is shown in this blue box. Again, I was just trying to show you the pickup of the electrons. This is only a portion of the molecule up at the top. The rest of the molecule is down here. But anyway, if you look right here, look at this hydrogen, and then over here there's been a pass-off of electrons right there. So NAD is being reduced. It is gaining electrons, reduction. And notice that there is an extra guy right there. Alright? So it's been reduced. It's now called NADH. That's its reduced name. NAD is its oxidized name. Okay. Here's kind of we're going to run through the nutrients. We're going to go carbs first, lipids second, proteins third. Alright, so carbs. Basically, this thing shows you how a cell, which is the big square guy, can use different kinds of molecules for aerobic respiration, because we're going to focus a lot on aerobic respiration here. So, we've got the fatty acids that can go right into aerobic respiration. By the way, the blue guy right here, mitochondrion. The glycerol molecule, remember a triglyceride fat is 3 fatty acids and glycerol. Glycerol can actually go into glycolysis. This is the glycolytic pathway right there. And then, of course, it goes into Krebs cycle in a mitochondrion. And then in the way of proteins, amino acids, remember they're broken down as amino acids, individual ones in the gut. Some of the amino acids, most of them actually, then go straight into the Krebs cycle. So again, your cells can use a variety of things. Types of carbs, remember, polysaccharides, disaccharides, monosaccharides. You're going to hear about all groups, all 3 groups. Starches, glycogen, those are your polys. Disaccharides, lactose, sucrose, maltose. Uses, carbs, number 1 source of energy. The easiest ones to break down. That's why your cells like to use them. The picture that you see down here, I'm just trying to show you differences between cellulose, which is a polysaccharide, and its cousins, starch and glycogen. Plants make starch, obviously. That's why we like to eat them. Meat has glycogen. You store glycogen in your own muscle. When you eat a steak, you're getting glycogen too. It's a complex carbohydrate or a polysac. Cellulose, on the other hand, close cousin, we can't digest it. We don't have an enzyme. We have no cellulase enzyme to break down cellulose. It's strings of molecules of glucose, but notice, although not important to know or to regurgitate on a test, different kind of bond. Okay, overall formula for cell respiration. You should already know this baby by now, and if you don't, it's time to learn it. You're going to get hit with it in micro if you haven't had it, if you haven't remembered it up to now. We're going to look at aerobic respiration. So here is the sum total of the formula. Now, let me make sure everybody understands what sum total means. You go on vacation, let's say for 2 weeks, and you go to New Zealand, North Island, South Island, and you're going bungee jumping, and you're going to go into a volcano, and you go on hikes, and your days are jam-packed. When you come back and somebody says to you, "What'd you do in New Zealand?" And instead of reeling off this whole long list, you say, "Well, we did all sorts of activities, physical activities, ate a lot of food, looked at Maoris, dancing, had a great time." That is the sum total of your trip. No details. Okay, same thing here. Sum total of aerobic respiration. No individual reactions right here. Okay, your starting material. Over here on the left, you've got your reactants. All right? There are only 2 that you see here. Here's a monosaccharide. This happens to be a glucose sugar. Now, You also have to have oxygen. Remember, this is going to be aerobic respiration. These two guys do not ever meet. They never come in contact with each other directly. We're going to talk more about that. Remember, this is a sum formula. Okay, over here, end products to the right. Water, CO2, ATP. That's what the cell wants. Now, look at the terms oxidation reduction. This oxygen right here, these oxygen molecules, and the glucose. The glucose molecule has all these hydrogens right here. Remember, hydrogens, electrons. The glucose is going to give these hydrogens away and is going to be oxidized to CO2. Look at CO2. There's no hydrogen there. Now, something's got to pick them up. Oxygen is going to be the electron acceptor. When it accepts hydrogens, it becomes reduced. Boom, water. Now, we're going to cover this in 3 steps. We're going to go glycolysis, then we're moving to Krebs cycle, then we're going to go to the electron transport chain. Okay, let's move. Glycolysis. Only about 8 or 9 steps. This is the baby glycolytic pathway right here that I don't want to go into in detail. All right. Okay. And basically what we're going to do is we're going to start with glucose right here. There it is. It's a 6-carbon molecule. You can see 6 balls, 6 carbons. And at the end we're going to end up with 2 pyruvic acids. There they are. Forget this down here. This is actually the transition step, or what can happen to pyruvic acid after glycolysis. So glycolysis actually stops right there. Okay, so anyway, let's start up here at glucose. Got this 6-carbon molecule. We're going to split it in 2. Nope, let me back up. First, let's do something else. We are going to stick a high energy phosphate at this end and at this end. And to do that, to get the high energy phosphate, we're going to have to input 2 ATP. We're going to strip a phosphate off of each ATP molecule, and we're going to stick one over here and one over there. And there you see it. Okay, there's that high energy phosphate on the left. Here's a high energy phosphate on the right. It's not important to know that this is a fructose 1,6-diphosphate. Fructose and glucose are the same formula, just a little bit different orientation of the molecule. It's called a diphosphate because it's got 2 phosphates, and one of the phosphates is on carbon 1 on the left. One of the phosphates is on carbon 6 on the right. See how easy this is? Okay, so now what we're going to do is we're going to split this guy in half, and we end up with 2 pretty even little molecules, glyceraldehyde phosphate and dihydroacetone phosphate, which actually gets converted to glyceraldehyde phosphate. So we end up actually with 2 of those guys right there. Now, the rest of this pathway goes on twice because you've got 2 of these little 3-carbon molecules. So one goes down the pathway, making all these little, items right here, and another one goes down the pathway. Okay, so we have this glyceraldehyde 3-phosphate. Notice NAD. That should tell you right away there's going to be a pass-off of electrons. I haven't mentioned it before. This is the first place it happens. So NAD is going to get 2 electrons, or hydrogen, from glyceraldehyde phosphate. That NAD now becomes reduced to NADH. This is a reduction reaction. Glyceraldehyde phosphate becomes reduced, ultimately, to pyruvic acid. Now, in addition, by the way, there are about 3 little reactions that are occurring here, although you don't see them. And one of the reactions requires the input of another phosphate. And also during this little set of reactions, there are 2 ATP made. Now, remember there were 2 glyceraldehyde phosphate molecules. So a second one comes down, continues this. We get more reduced NADH. We get 2 more ATP, and that's why there's a 4 right there. And we end up with pyruvic acid from each of the glyceraldehyde phosphates. That's glycolysis right there. That's it. Here's the 2 NADHs we made, 2 electron carriers. There's your 4 ATP. Now, we had to input 2. We got 4. So our net really is only 2. We've only made 2. We had to put 2 in to get 4. That's glycolysis. There's nothing else to say. Okay. So now, here are your pyruvic acids right there. Remember, you got 2. This is a mitochondrion right there in the center where this is happening. Now, glycolysis, it was going on in the cytoplasm. We're moving now into the mitochondrion where this is going to go on. This is called the transition step or the intermediate step. And in this process, we're going to convert pyruvic acid to this guy over here, acetyl coenzyme A. Notice that this part of pyruvic acid gets lopped off, and there is a sulfur combination coenzyme A stuck there instead. Now look at this COOH. That's a carboxylic acid group right there. Now, hydrogen tends to just wander off on its own, leaving a COO. And what does that COO become? Or is? It is CO2. Look right there. That's where the CO2 is coming from in aerobic respiration. Every time you lop off one of these carboxylic acid groups or carboxyl groups. Now, there's also a pass-off of electrons. You see NADH over here. So, pyruvic acid is going to pass off electron or hydrogen to NAD. NAD becomes reduced. Pyruvic acid becomes oxidized. That's the transition step. By the way, you can see that fatty acids and amino acids can enter in this step. They can be converted to acetyl coenzyme A and used by the cell for energy and continue on with aerobic respiration. Okay, so here we are in the Krebs cycle, and let me point out a couple of websites. I think I've got them open here. All right. Notice on my URL, actually go up to the search. I've got a Google search window right there. All right, look what I put, top right. I put in McGraw-Hill, that's a book pub, and Krebs. And when I put that in, let me do it. Here I am in Google, and the first thing that comes up is how the Krebs cycle works, and it's a animation. Boom. This is a really good little animation. I'm not going to play it, but it is. Okay, now if I put in glycolysis into that search window, leave in McGraw-Hill. McGraw-Hill's got the best animations, by the way. Here's one on how glycolysis works. Okay. If you went in and you put electron transport chain, oops, let me spell this correct, you will end up with one on electron transport chain. Okay. All right, so here we are over in the Krebs cycle. And again, we're inside of a mitochondrion. And here's our pyruvic acid right there from before, from at the end of glycolysis. And here is our transition step right there, making acetyl coenzyme A. Notice there's a little bit of CO2 we made. And here's some reduced NADH, which I haven't told you what we're going to do with yet. Here's our acetyl coenzyme A. Notice it's got two little carbons. Now, you can't see the balls, but there they are right there. Now, this is a cyclical set of chemical reactions. In glycolysis, I mentioned it was like 8 or 9 chemical reactions. They're linear, like if you just wrote them across a page. The first one on the left, the 9th reaction on the right, boom, 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 linear. Well, this is a cyclical reaction. So the last chemical reaction of the cycle is actually going to start off the first chemical reaction of the next cycle. All right, so here's our acetyl coenzyme A. It's going to react with a 4-carbon molecule from the last cycle, oxaloacetic acid. Okay, 4 carbons, 2 carbons, boom, you got 6 carbons. That molecule is called citric acid. There is your first reaction of the Krebs cycle. First. This is also called the citric acid cycle, by the way, because citric acid is the first thing made in the cycle. Right. Then we're going to tweak it a little bit. We're going to change the molecule around a little bit, change some of its side chains around. We have isocitric acid. Now, at this point, we are going to knock off a carboxyl group. Remember that COOH? Okay, and we go from a 6-carbon there to a 5-carbon there. That's where the carbon went to. And we're going to pass off electrons. We're going to give electrons to NAD, becomes NADH. Okay, so there's your oxidation, reduction, reaction right there. But now it's not called isocitric anymore. It's called ketoglutaric acid. The next reaction, okay, so there's reaction 2. Here's chemical reaction 3. Okay, here's going to be chemical reaction 4. We're going to pass off electrons again. Going to make another NADH. We're going to lop off another carboxyl group, CO2. Now we got a 4-carbon molecule right there. Okay, here's reaction number 5. At the end of 4 we had succinyl coenzyme A. Now the next reaction 5, we're going to make a little bit of ATP. There it is, right there. And we end up with succinic acid. Now for the next reaction, here's number 6. The carrier is not going to be NAD, it's going to be FAD, its cousin. And FAD becomes reduced to FADH2. Succinic acid gets oxidized to fumaric acid. We're going to tweak the molecule again and change side chain orientation, malic acid. Malic acid is going to give away a couple of electrons. NAD, now we got reduced to NADH. It's not called malic acid anymore. We're back to oxaloacetic acid at the end of the cycle. Cycle goes around again. Okay, now, why does it go around again? Remember, during glycolysis, when we split glucose, we ended up with 2 pyruvic acids. So, here's pyruvic acid number 1, goes through the cycle, making all these things all the way around. Then, the second pyruvic acid, number 2, does the same thing. Everything is doubled here because you have 2 pyruvic acids doing this. Okay, so here's the sum total of reaction here. Not that you need to balance it or even really know the reaction per se, except you need to know the stuff that's made. We make a whole bunch of reduced NADH. We make a little bit of FADH, its reduced cousin. We make a bunch of CO2. We make a little bit of something called GTP, which is the cousin of ATP. It actually gets converted to ATP. That's the Krebs cycle. In case you're wondering, well, who cares about all these other little molecules of isocitric, malic acid, stuff like that? The cell needs these other things to make other things. It's not like the aim here is just to make CO2 and ATP. These other molecules have a role in cell function. It's not important to sit down and memorize the Krebs cycle. It's not important to know isocitric acid or ketoglutaric. You need to understand what's going on. Where's the NADH coming from? That is, not what reaction's being made in, but how do you get it? What's it going to be used for? Well, we're going to talk about what it's used for in just a second. Okay. So let's talk about what it's used for. All right. Electron transport chain. You're looking at a mitochondrion. There it is right there. There it is right there. Mitochondrion's got all those folded insides. Two membranes. Okay, outer membrane, inner membrane. All right, inner membrane. Okay, now, if I take, let's see, get rid of that. Okay, so if I take a section right there, and I make a picture of that, that's what you're looking at right there, okay? Here's the outer membrane of the mitochondrion. Here's the inner membrane of the mitochondrion. All right, now embedded along this membrane, you've got some protein carriers. They're in purple, all right? You see the same thing over here. This is just showing you the different electron carriers and their drop-off in energy levels as the electrons are passed carrier to carrier. All right, so this is a bunch of electron carriers. They're passing off the electrons or hydrogens from one to the second to the third and so on. Okay, so we're going to start over here with NADH. Remember all those NADHs that we made in the Krebs cycle? Made about 6. Remember we also made some NADH in glycolysis. We made a couple of molecules in the transition step, plus we made a bunch in the Krebs cycle. All of those come in right here, all. All right, now, the inner membrane, all these proteins are embedded in the inner membrane. Okay, so first pass off. From NADH, it's going to go to flavomononucleotide. It's a variation of riboflavin, one of the B vitamins. By the way, NAD is nicotinic adenine dinucleotide, one of the B vitamins. B vitamins are really important in the production of these guys. Okay, so FMN now becomes reduced. It picks up electrons. NADH becomes oxidized. It becomes oxidized back to NAD. All right, now, this NAD is, of course, going to go back and pick up more electrons as the Krebs cycle continues and glycolysis continues and so on. All right, FMN now is going to pass electrons to quinone. This guy with a Q. He's a vitamin K derivative. Q is going to, or quinone, is going to pass off to cytochrome B. B, C, and A are cytochromes. They're iron-based proteins. Cytochrome B is going to pass off electrons to C. C is going to pass off to cytochrome A. By the way, look at the red route. That's the route of the electrons as they're being passed. Okay, now, cytochrome A is going to pass off to the last carrier. It's going to pass the electrons to the last carrier. And the last carrier is oxygen, O2. When oxygen picks up electrons, it becomes reduced to water. There's the reduction of oxygen. Let me go back to blue, I think. Okay, so here is your reduced molecule from the reduction of oxygen. Oxygen's the final electron acceptor. Okay, now remember there was one place where FAD was the prime carrier? Here's your FAD. It bypasses, the electrons don't get picked up by NAD at that point, they get picked up by FAD, and FAD just sends them down the chain further down. Okay, now look at these hydrogens out here. These hydrogens, they're not getting passed with electrons. Up to now they've been passed along, but now they're going to go somewhere else. They're going to go into this space right here in between the two membranes. They concentrate in this space, which means that you've got lots of hydrogen, protons, in the space, but you got low levels of hydrogen in here. That's a gradient, right? You got high gradient in this middle area. You've got a low level here. Everything wants to go from high to low to equilibrate. There's only one place where those little hydrogens can flow through. This guy right here is ATP synthase. It is a enzyme which, let me erase all that, uses the flow of hydrogens. As the hydrogens flow through this open channel, which is also an enzyme, that flow, movement of electrons, or movements of hydrogens that is, that energy from the flow is used by this enzyme to make ATP, and that's where most of the ATP in aerobic respiration is coming from. This thing that I just described for you is the electron transport chain. It's sometimes also called chemiosmosis, or the electron motive no, that's not what I meant to say. Let me get rid of that. The proton motive force. You've had this in general biology. You may know it by that name, and that's where most of the ATP is being made. Okay, so here's sum total of everything, showing you where all the ATP is made from. Here is the reduced NADH made in glycolysis. Here's reduced NADH that's made in the intermediate step, converting pyruvic acid to coenzyme to acetyl coenzyme A. And then here's the Krebs cycle. Here's all the reduced NADH and FADH, and notice all that stuff is involved in the electron transport chain that I just took you through. Here's the addition of all the ATP, not that you need to know it, and we get around 36, 38. Actually, they figured that it's not quite that efficient, so the number is a little bit lower. They've changed their minds, they being scientists. Okay, last, fermentation. Why would a cell do this? If it has no oxygen. Aerobic respiration requires oxygen, electron acceptor at the end. If you don't have oxygen, then there's two choices. A, cell dead. B, cell does something else. Two choices, that's it. It can't just stop and sit there, you know, like sitting in the road on the freeway. You can't do that. Okay, so the cell is going to do something else to make its ATP. It's going to do glycolysis. Glycolysis is the common pathway for both fermentation, which is anaerobic, and aerobic respiration. So here's the glycolytic pathway. You've seen it before. All right. Here's the end of glycolysis right here. So now we have one more step. What we're going to do is we are going to pass electrons from NADH back to pyruvic acid. Look at this NADH right here. It's reduced. We're going to give these babies back to pyruvic acid. Pyruvic acid now becomes reduced, and NADH becomes oxidized. And this molecule right here is now called lactic acid. If you look, wait a minute, let me trying to get the bar down here. Okay. So if you look at the at pyruvic acid here, you look at lactic acid right here, note a hydrogen right there and a hydrogen right there. They're coming from that guy right there. Okay, that's fermentation. That's it. Here's glycolysis, and then this is your fermentation step right there. And why is this important? You've had this in 2401. You covered it in muscle metabolism when you were talking about muscle fatigue. You talked about lactic acid accumulation. There should not be an E in lactic, but anyway. And you covered anaerobic respiration at that time, or anaerobic metabolism. Okay, I'm stopping right there.